learned from the biotechnology, as you would have learned from the previous chapter. बायोटेक्नोलॉजी जैसे कि आपने पिछले चैप्टर में पढ़ा इसेंशियली डील विद द इंडस्ट्रियल स्केल प्रोडक्शन ऑफ बायो bioharmaticals and biological using genetically modified microbes, fungi, plant and animals. The application of biotechnology include therapeutics, diagnostic, genetically modified crop for agriculture, processed food, bio remandation, waste treatment and energy producing. Three critical research areas of biotechnology are: first, providing the best catalyst in the form of improvement organism, usually a microbe or a pure enzyme. Second point, the creating optimal condition through engineered for a catalyst to act, and third point, downstream processing technology to purify the protein organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. मुख्य तौर पे प्रोडक्शन और हेल्थ पे biotechnology and biotechnology application in agriculture. Let us take a look at the three options that can be thought for increasing food production. First, agrochemical based agriculture. पहला agrochemical based, दूसरा organic agriculture, and third, genetically engineered crop based agriculture. The green revolution success in tripling the food supply, but it was not enough to feed the growing human population. Increased yield have partly been due to the use of improved crop varieties, but mainly due to the use of better management practice and use of agrochemicals, fertilizers and pesticides. However, for farmers in the developing world, agrochemicals are often too expensive, and further increase in yield with existing varieties are not possible using conventional breeding. As a traditional breeding techniques failed to keep pace with demand and to provide sufficiently fast and efficient system for crop improvement. Another technology called tissue culture got developed. What does the tissue culture mean? It was learned by the scientists during 1950 that whole plant would be regenerated from explant, that any part of plant take out and grown in the test tube under sterile condition in special nutrient media. This capacity to generate a whole plant from any cell explained is called totipotency. You will learn how to accomplish this in higher classes. Uh, it it is important to stress here that the nutrients medium must provide a carbon source such as a source, and also inorganic said vitamins, amino acid and growth regulators like auxins, cytokinins, etc. By application of this method, it is possible to achieve propagation of a large number of plants in very short duration. This method of producing thousands of plants through tissue culture is called micropropagation. Each of these plants will genetically identical to the original plant from which they were grown. That they are som- clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Try to visit a tissue culture laboratory with your teacher to better understand and appreciate the process. Another important application of the method is recovery of the healthy plant from disease plant. If the plant is infected with a virus, the meristem application and auxiliary is free of virus. Hence, one can remove the meristem and grow it into vitro obtained virus-free plant. Scientists have success in culturing meristem of banana, sugar cane, potato, etc. Scientists have even isolated single cells from the plant and after digesting their cell walls have been able to isolate naked protoplast surrounded by the plasma membrane. Isolated protoplast from two different varieties of plant, each having a desirable character, can be fused to get hybrid protoplast, which can be further grown to form a new plant. These hybrids are called somatic hybrid. While the process while the process is called somatic hybridization, imagine a situation when a protoplast of tomato is fused with that of potato, and then they are grown to form new hybrid plant combining tomato and potato characteristic. Well, this has been achieved result in formation of potato. Unfortunately, the plant did not have all the desirable combination of characteristic for its commercial utilization. Is there any alternative path that our understanding of genetics can show that farmers may obtain maximum yield from their field? Is is there a way to minimize the use of fertilizers and chemicals so that their harmful effect on the environment are reduced? Use of genetically modified crop is possible solution. Plant bacteria, fungi and animals those genetic have been alternated by the manipulating called genetically modified organism. GMO, genetic modified organisms. Plants have been useful in many ways. Genetic modification has first made crop more tolerant to abiotic stress, cold, drought, heat. Second point, reduce reliance on chemical pesticide, pre-resistance crop. Third, help to reduce post-harvesting losses. Fourth, increase efficiency of mineral uses by plant. This prevent early exhaustion of fertility of soil. Fifth, enhance nutritional value of goods. Example, golden rice, vitamin A enriched rice. In addition to this uses, GM has been used to create tailor-made plant to supply alternative resources to industries in the form of stress, fuel and parth- parthem- parthimicutals. Some of the applications of biotechnology in agriculture that you will study in detail are the production of pest resistance plant which decrease the amount of pesticides used. Bt toxins is produced by a bacterium called Bucus thuringienus, Bt for short. Bucus thuringienus toxin genes has been cloned from the bacteria and been expressed in the plant to provide resistance to insect without need for insecticides. In, if, in affected create of biopesticides, examples are Bt corn, rice, tomato, potato and soybean. It is the Bt cotton, some strains of Bucus thuringienus produce protein that kills certain insects such as Lepidopterans, tobacco, budworms, armworms, coleoptera, beetles, and dipaternis, flies, mosquitoes. Thuringienus forms protein crystals during a particular phase of their growth. These crystals contain a toxic incidental protein. Why does the toxins not kill mucus? Actually, the actually the actually the mucus thuringienus toxins proteins exist as inactive prote- pro- proteins, but once an inject the inactive toxin, it is converted into active forms of toxin due to the alkaline pH of gut, which soluble the crystal. The active toxins bind to the surface of midgut epithelial cell and create pore that causes swelling and lysis and eventually of the insect. भाई ये भाई आगे स्कैनर क्यों रखा है? अरे यार स्कैनर उस पर लिखा है भाई। भाई सब्सक्राइब करो। Specific Bt toxin genes were isolated from Bucus thuringiensis and incorporated into the several crops plants such as cotton. Figure 10.1. Figure 10.1 cotton balls A destroyed by bollworms. A destroyed by a bollworms and B a full mature cotton ball. The choice of gene depend upon the crop and targeted pest, as most Bt toxins are insect group specified. The toxins is coded by a gene cry cry named cry, and there are a number of them. For example, the protein encoded by the genes cry IAC and cry IIB control the cotton ball worms. That of cry IAB control the corn ball. Pest resistance plant. Pest resistance plants. Several nematodes, parasites, a wide variety of plant and animals, including human beings, or nematode milieu in Inocogita infect the root of tobacco plant and causes a great reduction in yield. A novel strategy was adopted to prevent these infections, which was based on the process of RNA interface. RNA interface. RNAi. RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA messenger ribose nucleic acid due to a complementary dsRNA molecule that bind to prevent translation of the mRNA. Silencing the source of the complementary RNA could be form an infection by virus having RNA genomes or mobile genetic element transportions that replicate via RNA intermediate using agrobacterium vectors. Limited specific genes were introduced into the host plant. Figure 10.2. Figure 10.2. Figure 10.2 host plant generated dsRNA triggers protection against nematoids in infestation. First, a root of typical controlled plant. Root of typical control plant. B transgenic plant root. 5 days after deliberated infection of nematode, but protected through novel mechanism. Protected through novel mechanism. The introduction of DNA, जो introduction of DNA was such that it was produced both sense and antisense RNA in the host cells. These two RNAs being complementary to each other formed a double standard dsRNA that initiate RNAi interface and such silence the specific mRNA messenger ribose nucleic acid of the nematoid. The consequence was the parasite could not survive in a transgenic host expressing specific interface RNA. The transgenic plant therefore got itself protected from the parasite. Figure 10.2. Hmm. Biotechnology and its application. Page number 181. 10.2 Biotechnology technology application in medicine. The recombinant DNA technologies proceed have met immense impact immense impact immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drug. Further, the recombination ther- therapeutics do not introduce unwanted immunological response as is common in the case of similar product isolated from non-human resources. And about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. 10.21 genetic engineered insulin. Management of adult one onset diabetes is possible by taking insulin at the regular time intervals. What would a diabetes patient do if enough humans insulin was not available? If you discuss this, you will soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from another animal just as effective as that secreted by the human body itself, and would it not elicit in an immune response in the human body? Now imagine if bacteria were available that could make human insulin. Suddenly, the whole process becomes so simple. You can easily grow a large quantity of the bacteria and make as much as much insulin as you need. Think about whether the insulin can be orally administered to diabetes people or not. Why? Insulin used for diabetes was earlier extracted from the pancreas of the celebrated cattle and pigs. Insulin from an animal source thought causes some patient to develop allergy or other uh, type of reaction to the foreign protein. Insulin consists of two short polypeptide chain A and that are linked together by the dis 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 dispulted breeze. Figure 10.3. Biology is number 182. In mammals, including humans, insulin is synthesized as a pro-hormones like a pro-enzyme. The pro-hormones also needed to proceed before it become a full mature and functional hormones. which contain an extra stretch called C peptide, जिसे कि हम C peptide कहते हैं. Uh, this C peptide is not presented in the mature insulin and is removed during the maturation into insulin. The main challenge for the production of insulin using rDNA technique was getting insulin assembled into a mature from 1983. An American company, prepared two DNA sequence corresponding A and B chain of human insulin and introduced them in a plasmid of E. coli to produce insulin. Chain A and chain B were produced separately, extracted and combined by creating this 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 ulfid bond to form human insulin. Figure 10.3. Maturation of pro-insulin into an insulin simplified. First, pro-insulin SSSS A peptide SSS insulin B plus B peptide. 10.22 gene therapy. 10.22 gene therapy. If a person is born with a hereditary disease, can corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Genetic therapy is a collection of methods that allow correct gene defect that has been diagnostic in a child embryo. Here, genes genes are inserted into a person's cell and tissue to treat a disease. Correction of a genetic defect involves the delivery of normal gene into the individual or embryo take over the function of an egg and compensate for the non-functional gene. The first clinical gene therapy was given in 1909. 4-year-old girl 4-year-old girl with adenosine diamonds ADA deficiency. This enzyme crucial for the human system immune system to function. The disorder is causes due to the deletion of the gene for adenosine diamonds. In some children, adenosine diamonds deficiency can be cured by bone marrow translation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to parent by injection. Problem with bond of these approaches are that are not completed curative. As a first step toward gene therapy, lymphocytes from the blood of the parent are grown in a culture outside the body. A functional ADA, adenosine diamines cDNA, using a retroviral vector, is then introduced into this limo oxide, which are subsequently returned to the patient. However, as these cells are not immortal, the parental required periodic infusions of such genetically engineered lipooxides. However, if the genetic isolated from marrow cells producing ADA is introduced into cell at early embryonic stage, it could be a permanent cure. 10.10 10.23 molecular diagnosis. You know that if and you know that आप जानते हैं, you know that effective treatment of disease, जो effective treatment of disease है, early diagnosis and understand it patho pathology is very important. Using a conventional method of diagnosis, system and urine analysis, etc. Early deletion is biotechnology and its application not possible. Recombinant DNA technology, polymers, chain reactions, PCR and enzymes linked immunosorbent एस 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 से ई एस इसे शॉर्ट में कहते हैं ई एल एस से आर are some of the techniques that serve the pure purpose of early diagnostic. Presence of pathogens, bacteria, virus is not normal suspected. Only when the pathogens has produced disease symptoms. By this time, the concentration of pathogen is already very high in body. However, very low concentration of bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected via amplifications and their nucleic acid by PCR. Can you explain how PCR detect very low amount of DNA? PCR is now routinely used to detect HIV in suspected AIDS patients. It is it is being used to detect mutations in genes in, in suspected cancer parents took. It is a powerful technique to identify many other genetic disorders. A single transit DNA or RNA targeted with radioactive molecules probe is always to hybridize to its complementary DNA in a clone of cell followed by deletion using autoradiography. Cloning. The cloning have the mutated gene which uh, will hence not appear on the photographic film because the probe will not have complementary with the mutated gene. ELISA is based on the principle of the anti-body interaction. Infection by pathogen can be detected by the presence of antigens, protein, glycoprotein, etc., or by detecting the antibodies synthesized against the pathogens. 10.3 transgenic animals. Deficiency है भाई. Okay brother, okay. ओके ठीक है भाई. 1909 क्लिनिकल थेरेपी एडिनोसिन. इसमें सबसे पहले 1909 में 4 साल की लड़की को एडिनोसिन डायमिन्स डिफिशिएंसी दिया गया था. दैट इज क्रूशियल फॉर इम्यून सिस्टम ऑफ फंक्शन. उसमें उसके उसके बीमारी को निकाल दिया गया था काट करके. उसमें दूसरा जीन को अटैच कर दिया गया था. अच्छा भाई तुम हो कि नहीं हो? चलो ठीक है. animals that have animals that have had their DNA to process and express an extra foreign genes are known as transgenic animals. The transgenic rats, rabbit, pig, sheep, cow, and fish have been produced all through over 19% of all existing transgenic animals are mice. Why are these animals being produced? However, how can many benefit from such modifications? Let us try and explore some of the common reasons. Normal physiology and development. Transgenic animals can be simply simplifically designed to allow the study of how genes are regulated and how they affect the normal function of body and its development. Example, study of complex factor involved in growth such as insulin like growth factor by introducing genes from other species that alter the formation of these factors and studying the biological effect that result information is obtained about the biological role of the factor in the body. So, and study of disease. Second point, many transgenic animals are designed to increase our understanding of how genes contribute to development of disease. These are especially made to serve as a model for humans disease so that investigation of new treatment for disease is possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, thyroiditis, arthritis, and Alzheimer's. 184 biology. बैट बायोलॉजिकल प्रोडक्ट क्या होता है इसके बारे में जानेंगे. Biological product medicine required to treat certain human disease can contain biological product. Such products are often expensive to make. The transgenic animals that produce useful biological products can be created by the introduction of the proportion of DNA or genes which code for a particular product, such as a human protein alpha A and antitrypsins used to treat em, emphysema. Similar attempts are being made for treatment of phalancantinuria, PKU, phalancantinuria, PKU, and 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 cystic fibrosis. In 1979, the first transgenic cow, Rosie, produced human protein enriched milk 2.4 gram 2.4 gram per liter. The milk contained humans alpha like the milk content alpha lactamin was neutrally a more balanced product for humans babies than natural cow milk. Fourth, vaccine safety. Transgenic mice are being developed for use in the testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccines. If successful and found to reliable, it could replace the use of monkeys to test safety of batches of vaccine. Fifth, chemical safety testing. This is known as a toxicity safety testing. The procedure is the same as that used for testing toxicity of drugs. The transgenic animals are made that carry genes that carry genes which make them more sensitive to toxin substance than non-transgenic animals. They are then exposed to toxic substance and the effects studied. The toxicity testing in such animals will allow us to obtain result in less time. 10.4 ethical issue. The manipulation of living organism by the human race cannot race cannot go on any further without regulation. Some ethical standards are required to evaluate the mortality of all human activities that might help or harm living organisms. Going beyond the mortality of such issues, the biological significance of use of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations GE such as GEAC, Genetic Engineering Approval Commit, which will make decisions regarding the validity of GM research and the safety of the introducing GM or organisms of public service. The modification use of living organism of for such for public service as food and medicine sources, for example, has also created problems with parent granted for some. There is a growing public anger that certain companies are being guaranteed patents for products and technologies that make use of the genetic material, plant, and other biological resources that have long been identified, developed, and used by farmers indigenous people of a species region's country. 1.85 one page number 185, biotechnology and its applications. Ra- एक second. राइस इज एन इम्पॉर्टेंट फूड चैन. जो राइस है वो बहुत महत्वपूर्ण फूड चैन है. द प्रेजेंस ऑफ विच गोज बैक thousands of years. Asia agriculture history. There are an estimated 2 lakh varieties of rice in India. Alone, the diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and 27 documented varieties of basmati are grown in India. There is a reference to basmati in ancient texts, flores, and poetry as it has been grown for centuries. In 1997, in 1997, a company got patent right on basmati rice through the US Patent and Trademark Trademark Office. This allowed the company to sell a new variety of basmati in US and abroad. The new variety of basmati has actually been diverted from the Indian farmers variety. Indian basmati was crossed with semi-dark variety and claimed as a invented or uh, novelty. The patented extend to functional equivalent, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent uses product and process based on Indian tradition, herbal medicine, example turmeric, neem. If we are not vigilant and we do not immediately counter this patent application, our country's individuals may encase our rich legacy, and we may not be able to do anything about it. Biopiracy. Biopiracy. It is a term used to refer to the use of bioresource by multinational companies and other organisms without proper authorization from the countries and people concerned, without com- compensatory payment. Most of the industrialist nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloping world is rich in biodiversity and traditional knowledge related to bioresources. The traditional knowledge related to the bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure. During their commercialization, there has been growing realization of the justice in aquatic compensations and benefits sharing between developed and developing countries. Therefore, some nations are developing laws of prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second admonitions of the Indian Patents Bill that take such issue into considerations, including patent terms, emergency provisions, and research and development initiatives. खत्म वाह समरी बायोलॉजी समरी Biotechnology has given to humans several useful products by using microbes, plant, animals, and their metabolic machinery. Recombinant DNA technology has made it possible to engineer microbes, plant, and animals such that they have novel capabilities. Genetically modified organisms has been created by using method others than natural method to transfer one or more genes for from one organism to from one organism to another genetically using techniques such as recombination DNA technology. GM plants have been useful in increasing crop yield, reduces post harvest loss, and make crop more tolerant of stresses. There are several GM crop plant with improved nutritional value of food and reduce the reliance or on chemical pesticide. Pest resistance crop recombinant DNA technological process have made immense impact in the area of healthcare by enabling a mass production of safe and more effective thera- therapeutics. Since the recombinant therapeutics are identical to human proteins, they do not induce unwanted immunological response and are free from risk of infection as was observed in the case of similar product isolated from non-human source. Human insulin is made in bacteria. Its structure is absolutely identical to that of the natural molecules. Diagnostic, transgenic animals are also used to understand how genes contribute to the development of disease by serving as a models for human disease such as a cancer, cytics, fibrosis, rheumatoid arthritis, and Alzheimer's. Gene therapy is the insertion of genes into an individual cell and tissue to treat disease, especially hereditary disease. It does not so by replicating a defective mutant allele with a functional one or gene targeting which involves gene amplification virus that attacked their host and introduced their genetic material into the host cell as a part of their application cycles are used as a vector of transfer health genes or more recently portions of genes. The current interest in the manipulation of microbes, plant and animals has raised serious ethical questions. डिफेंसी है भाई